Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntazir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is glycolysis. Glycolysis is carried out for obtaining energy in the living organisms. Living organisms desire and crave for a regular and continual supply of energy to perform biological work and all the body functions. The energy rich molecule ATP usually arrange this energy. All the organisms produce and create energy that is in the form of ATP by the breakdown and cracking up of organic nutrients such as carbohydrates, lipids, proteins, and nucleic acids. This life-saver discharged and released energy is used to combine ADP with phosphate to form ATP. In the living organisms, the breakdown of organic nutrients such as glucose takes place in a step-by-step series of chemical reactions that is known as glycolysis. The word glycolysis is the combination of two words, two profabs. The profab glyco means glucose and the profab lysis means splitting and breakdown. So the glycolysis is a systematic metabolic pathway that involves systematic disruption of glucose to produce energy. We can say that glycolysis takes place in the cytosol of the cells. So, the confab glycolysis means breakdown and splitting of sugar such as glucose. In living organisms, it is approximately a general method, a general pathway that converts, changes glucose to pyruvic acid. This pyruvic acid is the end product of glycolysis that is further broken down either in the presence of or in the absence of free oxygen. So there are two possibilities. The first one is aerobic respiration and the second is fermentation or anaerobic respiration. The definition of aerobic respiration is that when end product of glycolysis, that is pyruvic acid, is further broken down in the presence of free oxygen, then the process will be known as aerobic respiration. Whereas when pyruvic acid is further broken down into smaller units in the absence of free oxygen, then this process will be known as fermentation, also called anaerobic respiration. Glycolysis and fermentation, both these processes occur in the cytoplasm because during eukaryotic cell evolution, the enzymes that catalyzed each reaction remained dissolved in the cytoplasm and did not localize in membranous organelles. So the origin of glycolysis and fermentation took place before the evolution of complex organelles. These reactions are also older in an evolutionary point of view than aerobic respiration because they could have occurred in the earliest primitive environment of the earth before the atmosphere contained free oxygen. Glucose, that is a 6-carbon sugar, is broken down into two molecules of 3-carbon sugar, that is a glyceraldehyde 3-phosphate. These two molecules of smaller sugars are oxidized. Their remaining atoms are reorganized to form two molecules of pyruvate. The catabolic pathway of glycolysis consists of many steps, each progressed by a specific enzyme that we will see soon. The enzymatically catalyzed reactions in glycolysis can be summarized as glucose to ADP to phosphate and to NADH that will produce two molecules of pyruvic acid, two molecules of ATP and two molecules of NADH. Now we will see the step-by-step reactions that are involved in glycolysis. For the glycolysis process happens in two phases. During the first phase, glucose is converted into two molecules of glyceraldehyde 3-phosphate. Although this conversion involves the addition of phosphate, first phase actually uses energy in the form of ATP instead of producing it. In the second phase, The glyceraldehyde 3-phosphate is converted into pyruvate. During this phase, the energy is actually produced. For its understanding, first of all, we take glucose. Glucose is converted into glucose 6-phosphate in the presence of an enzyme hexokinase. The word hexo means 6, representing the 6-carbon compound glucose, and the kinases are group of enzymes that catalyze the phosphorylation reactions. So the enzyme hexokinase transfer a molecule of phosphate from ATP to glucose and converts glucose to glucose 6-phosphate. In this reaction, ATP is changed into ADP. Hence, it utilizes one molecule of ATP. An ATP molecule is invested in this first step. Glucose 6-phosphate is reorganized to form its isomer fructose 6-phosphate in the presence of an enzyme phosphoglucoisomerase. During this second step, ATP molecule is not invested. During the third step, fructose 6-phosphate that reacts with second molecule of ATP, that is addition of another phosphate group to the carbon number first of fructose ring, is converted into 1,6-diphosphate in the presence of an enzyme phosphofructokinase, another kinase enzyme which transfer a phosphate group from ATP to fructose 6-phosphate. Again, ATP is converted into ADP. Hence, ATP is used in this reaction. Fructose 1,6-diphosphate is also known as hexose diphosphate. Fructose 1,6-diphosphate is also converted into two molecules that are 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. In the presence of an enzyme aldolase, an equilibrium is established between these two compounds by interconverting into one another in the presence of an enzyme isomerase. Students, keep this important point in your mind that first and third step, during which two ATP molecules are used as a starter energy to convert glucose to fructose 1,6-diphosphate, are energy requiring steps of glycolysis. Because in these steps, two ATP molecules are invested. All these reactions are composed of first phase of glycolysis, and you can see energy is actually consumed in this first phase in the form of two ATP molecules. Now we will utilize 3-phosphoglyceraldehyde at a faster rate, but when this compound is present in its very deficient amount, then dihydroxyacetone phosphate is converted into 3-phosphoglyceraldehyde, which is further processed during glycolysis. 3-phosphoglyceraldehyde is initially oxidized by NADH, and in this reaction, nicotinamide adenine dinucleotide, that is shortly known as NADH, that is a coenzyme and is also reduced to NADH and further utilized to electron transport chain to produce more and more energy. This reaction also uses a molecule of inorganic phosphate. The inorganic phosphate that is present in the cytoplasm combines with it. The glyceraldehyde 3-phosphate is further utilized in the glycolytic process. An equilibrium of this isomerization reaction is always towards the glyceraldehyde 3-phosphate. Also keep in mind that every molecule of glucose is split into two molecules of pyruvate. So every reaction of the second phase takes place twice and produces twice of products. And then 3-phosphoglyceraldehyde is converted into 1,3-diphosphoglyceric acid or 1,3-diphosphoglycerate in the presence of an enzyme triose phosphate dehydrogenase. That is a fifth step. During the sixth step, 1,3-diphosphoglyceric acid is converted into 3-phosphoglyceric acid or 3-phosphoglycerate in the presence of an enzyme phosphoglycerokinase. Phosphoglycerate kinase enzyme transfers the phosphate group at the first position of phosphoglycerate to adenosine diphosphate. So an ATP molecule is generated, but collectively two ATP molecules are obtained because second phase happens twice. This step releases two ATP molecules. During the seventh step, 3-phosphoglyceric acid or 3-phosphoglycerate is converted into 2-phosphoglyceric acid or 2-phosphoglycerate in the presence of an enzyme phosphoglyceromutase. Next one is eighth step, during which 2-phosphoglyceric acid or 2-phosphoglycerate is converted into phosphoenol pyruvic acid or phosphoenol pyruvate in the presence of enolase enzyme. And magnesium is also utilized in this eighth step. During the ninth step, ultimately, phosphoenol pyruvic acid or phosphoenol pyruvate is converted into pyruvic acid or pyruvate in the presence of an enzyme pyruvate kinase. This last step also generates two more ATP molecules. This all composes the second phase of glycolysis, and you can see energy is produced from the second phase of glycolysis. Let's first consider the net reaction of glycolysis, and then we will understand it briefly. As we have one molecule of 6 carbon glucose that splits into two molecules of 3 carbon compounds pyruvate, there is a net utilization of only 2 ADPs, since 4 ADPs are obtained during the second phase, out of which 2 ADPs are utilized during the first phase. So there is a net use of 2 ADPs and 2 molecules of inorganic phosphate in the form of phosphate that are utilized during the fifth step. 2 molecules of nicotinamide adenine dinucleotides ions are reduced to 2 NADH molecules. These are reduced during the conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So this is the overall diagrammatic picture of the glycolysis. As we have known that two ATP molecules are generated during the conversion of 1,3-diphosphoglycerate to 3-phosphoglycerate, and two more ATP molecules are obtained during the conversion of phosphoenol pyruvate to pyruvate, the end product of glycolysis. So students, in addition to 4 ATP molecules that are obtained during glycolysis, energy-rich compound NADH2 is also obtained during glycolysis, that is used to make ATP during oxidative phosphorylation. Finally, we can say that for each molecule of glucose that takes part in glycolytic pathway, 4 molecules of ATP are obtained. Although 2 ATP molecules are used to rearrange the next glucose molecule to form a new 6 carbon compound. So the net energy obtained from glycolysis process per glucose molecule is 2 ATP molecules along with 2 NADH. So students, it is very unique and important way to understand glycolysis metabolic pathway. You have to memorize only 10 names of those products that are produced during step-by-step reactions, and also 10 names of those enzymes that are used during each step at your fingertips. As after the participate of glucose, each product is converted to next product in the presence of that special correspondence enzyme. So by memorizing 10 names of enzymes and 10 names of products, you can easily write this process in your own words. It is not a difficult method, but it is very easy. But only you should memorize the names of products and enzyme. It is very important topic that often comes in annual exams. When I was the candidate of FSC and BSc examination papers, then I had also received this question in my exams. So I can say that it is very important topic. Understand it very well and make those actions, those works that are benefited for your country and the future of your country. For it, first of all, your responsibility is that get the education eagerly, bona fides, and not only operously but by hard working in that field in which you want. Today it is my encouragement for you. At the end, many thanks for watching this your video.